So, students of PT third sem, today I'm going to discuss about the other part of alternative splicing, which are the types of alternative splicing that we have not mentioned in the previous lecture. So, let's get started now. So, about the modes of alternative splicing. So, let us see about the modes of alternative splicing. What it has been mentioned here. So there are various modes of alternative splicing, like here it is saying like exon skipping. It is the normal procedure for splicing process where you can see the E that is referred to here. These are the exons given as Roman 1, 2, 3, and 4, whereas the I refers to the introns. Okay, so what says about the exon skipping here that uh, this mechanism, this mechanism called exon skipping, it is also known as cassette exon. It is also called as cassette exon. In this case, an exon may be spliced out of the primary mRNA. Okay, from the primary transcript, that is the mRNA that we get from transcription process, one or two exons may be spliced out. Generally, we know that introns are actually spliced out and exons are joined in a normal way, but sometimes what we call as exon skipping, where due to the presence of certain splice sites within the within the exon, it may leads to skipping of that particular exon to make a variant transcript, to make a variant mature mRNA, so that we can get an alternative protein from that mRNA. Okay, so it is saying that's why that this mechanism is also called as cassette exon. In this case, an exon may be spliced out of the primary transcript. That means from the primary mRNA, one or two exons may be spliced out. Best example of exon skipping is the DSX gene found in the Drosophila melanogaster, which is fruit fly. Okay, so the best example of exon skipping is your DSX gene, which is found in the fruit fly, that is Drosophila melanogaster. Normally, males, the DSX gene have a series of exons. Normally, males have exon 1, 2, 3, 5, and 6. The exon number 4 is missing within the male DSX gene, while the female have 1, 2, 3, 4 exon for that DSX gene. The 5, 6 exon is missing in female, whereas in male, exon number 4 is missing. That means the exon number 4 in male has been skipped out, whereas in female, exon number 5 and 6 has been skipped out. Okay, actually, it is saying why this happens, why exon skipping happens. For that, the reason has been mentioned here through an help of an example. Actually, a polyadenylation signal is present in the exon 4. So within the males, within the males of Drosophila melanogaster, the DSX gene, which is found in the males, the DSX gene that is found in the males, they have the exons, right? So the exon number 4 of that DSX gene in males, they have a polyadenylation signal within them, within the exon number 4, and that causes the transcription process to stop at that point. Exon number 4, on the other hand, in females is added because of one of the activators present only in female and not in males. So if I say in a stepwise manner, it is saying that in I have said that the DSX gene that is found in the male, their exon number 4 is missing. Exon number 4 is missing. Whereas in female, they have the exon number 1, 2, 3, 4 for that DSX gene, but exon number 5 and 6 is missing. Why? Because in males, their exon number 4 consists of a polyadenylation signal that causes the transcription machinery to stop at that point. Okay. So, and in case of female, the exon number 4 is added because exon number 4 is added to the female because they have an activator sequence that is required, that is present only in female for the expression of exon number 4 within the during the splicing process. Okay, and not, it is not found in males actually. So exon skipping is the most common mode of alternative splicing in the mammalian pre-mRNA. So let us see the exon skipping here. Here it has been mentioned like exon number 1 and 4 may be skipped out together, only 2 and 3 can remain, or it may occur that exon number 1 and 2 will retain, the rest of them can be skipped out. Okay, or the 4 of them may be retained together, introns may be removed out in that way. Okay, so this is a example of exon skipping, where again I repeat, in exon skipping an exon may be spliced out from the primary mRNA. Okay, and best example for exon skipping is DSX gene, which is found in Drosophila melanogaster. Male do not have the exon number 4 for that DSX gene, while the female have the 4 exons, one exon number 1, 2, 3, 4, but they don't have exon number 5 and exon number 6. Okay. The next type of alternative splicing is your mutually exclusive exons. Mutually exclusive exons. What does it mean? What does it mean in case of mutually exclusive exons? It is saying here that in mutually exclusive exon mode, one of the two exons is retained. Here the word is not able to justify. It is saying retained, retained. Okay, so it is saying that in mutually exclusive exon, one of the two exon is retained in the mRNA after splicing, but not the both. So it is an, what can I say, that in mutually exclusive exon, one of the two exon will be retained in the mRNA, not both. Both all the there are some exons which retain only in the mRNA. Both of them will not remain in the mRNA. Either this or that. Just like I can say, for with the help of example, I can say. So here, if you see in the figure, in this page, okay, what does it says? In this figure, it says, in this figure that you can see in mutually exclusive exon, it is saying that exon number 1 can be retained with exon number 3. That means after splicing, exon number 1 will combine with exon number 3, or exon number 1, or this even can also occur, exon number 1 can retained with exon number 2, but 3 of them will not remain together. Exon number 1, 2, 3 will not remain together. Either exon number 1 will retain with exon number 3, or exon number 1 will retain with exon number 2. Okay. Similarly, this arrow mark shows the thing. Similarly, exon number 2, if I use different color, similarly exon number 2 can retain with exon number 4, right? Or exon number 2 can retain with exon number 1. So exon number 2 can retain with exon number 4 to make a different spliced product of mRNA, or exon number 2 can retain with exon number 1. Okay, for that is a mistake here. Okay, 1 and 2. That means either exon number 2 retain with exon number 4, or exon number 2 can retain with exon number 1. Three of them will not remain together. Three of them will not remain together. 1, 2, 4 will not remain, just like the previous case. I hope you have understood. So in short, we can say that exon number 1 and 4 can retain with either with the exon 2 and 3. So in short words, we can say that this exon number 1 and exon number 4 can retain with either exon 2 or exon 3. All of them will not remain together. So this is called as mutually exclusive exon. So again I repeat, in mutually exclusive exon, one of the two exon will be retained, will be remain in the mRNA transcript after splicing, but not all of them, but not the both. Like in this figure you can see that exon number 1 can retain with exon number 3 to make this product, exon number 1 and 3, or exon number 1 can retain with exon number 2 to make exon number 1 and 2. But all of the three, exon number 1, 2, 3, will not remain after splicing. That's why it says mutually exclusive. Similarly, if I use a different color, similarly exon number 2, exon number 2 can be retained with exon number 4 to make exon number 2 and 4, or exon number 2 can retained with exon number 1. Okay, so the combination may be, what are the combinations that may occur from this mRNA? From this mRNA transcript, we can get this combination like this. Okay, so from this we can get a combination like this. We can get either 1, 3, 4. You can see here this has been joined with this, so exon number 1 can combine with 3 and combine with 4 here. Okay, this is one mRNA product after splicing. Okay, second, we can use a different color here. Exon number 1 can combine with exon number 2 and then exon number 4. So exon number 1 to 4 is the second way which can we get alternative splicing product. Okay, can I see the third way here? Third way, 1, 3, 4 I have said, 1, 2, 4 I have said. Okay, so these two products can be involved. Okay, so these two combinations we can get, okay, through these mutually exclusive exons. The best example for mutually exclusive exon is Timothy syndrome. Now it is saying mentioned here that in Timothy syndrome, the alternate forms of these two exon, which is called as 8 and 8A, can lead to a different symptoms of this disease, which causes disruption of the calcium homeostasis that is needed for muscle contraction. So there is a syndrome called Timothy syndrome. Why this occurs, I will send you the link, uh, send you, or in detail I will talk to you. Okay, just for knowledge, in Timothy syndrome, the gene responsible for that syndrome occurs having two types of exon, 8 and 8A. Okay, 8 and 8A. All the patients will not have both of these exons, either 8 or 8A. Okay, so in Timothy syndrome, these two exons can lead to different symptoms of the disease. Those patients who have this 8 exon number 8, they have a different syndrome, the symptoms. Those who have 8A, they have a different symptoms of the disease, which cause disruption of the calcium homeostasis, which is needed for your muscle contraction. We all know that for muscle contraction, calcium is very important signal molecule here. Okay, to maintain then for muscle contraction and relaxation, we need calcium. Obviously, calcium channels are also involved in your sarcoplasmic reticulum, that we know. So, but according to the exon, mutually exclusive exon, if I say in that point of view, both exons cannot exist in patient. Both the 8 and exon number 8A will not exist together. Either 8 will remain, exon number 8, or 8A will be remain. So only one of them is transcribed, though both are present in the same gene. So within the same gene, there is a exon 8 present and exon 8A is also present. But within the patients who are suffering from Timothy syndrome, either they will have the 8 number exon or they will have 8A exon. Okay. So this is an example of your alternative splicing. Um, the types of alternative splicing, which is called mutually exclusive exons. Okay, I think you have understood this part about the mutually exclusive exons. The best example is Timothy syndrome, where the patients will have either exon number 8 or they will have exon 8A. Not both of the exons will be present, but both of them leads to the disruption of your calcium homeostasis, which is needed for your muscle contraction and relaxation. Okay, now let us discuss about the third type of alternative splicing, which is called as alternative 5 prime alternative 3 prime acceptor site. Don't get confused. Well, acceptor site is your 3 prime splice site. In previous words, in previous days, we call the 3 prime splice site as an acceptor site. Remember the acceptor site? I hope you have remembered the acceptor site. Acceptor site is the 3 prime splice site within the intron exon boundary. So if this is an exon, if this is another exon, I am drawing the exons by purple color. Okay, and this is your 5 prime, this is your 3 prime, and a blank space is the intron, suppose. So towards the 5 prime in is the 5 prime splice site, and towards the 3 prime in is the 3 prime splice site of that intron. Okay, we are talking about this one, the 3 prime splice site, which is called as acceptor site. So what we call as 5 prime splice site, 5 prime splice site is called as donor site. Okay, so we'll come into that next later on. So let us see, it is saying here about alternative 3 prime acceptor site. So what does alternative means? Alternative means another 3 prime acceptor site. Well, there is a 3 prime splice site present in every intron, but sometimes alternative 3 prime splice site may also found, which may leads to skipping of that exon. So what does it say? So it is saying here that here exons have a alternative 3' splice site, which is called as acceptor site, so that they can bind with alternative way to the 5' splice site of exon number 1. Here it is shown. Again I repeat, here in alternative 3' acceptor site, which is called as 3' splice site, the exons having alternative 3' splice site, thus they can bind with alternative way to the 5' splice site of exon number 1. Here it has been shown. So exon number 1, so if I zoom it, you can able to understand what I am trying to say. Suppose there is a 5, so this is the 5', this is the 3', and this is the 5' splice site. This is the 3', this is the 3' splice site. There is an alternative 3' splice site here. Okay, so two cases can occur, either exon 1 during the splicing process, as they have a normal 5' splice site and 3' splice site, it can be skipped out and the intron can be skipped out and exon 1 can join with exon number 2. Or another event can occur. That event is, okay, exon number 2 and 3, okay. Another event can occur. The next event is they have an 3' splice site, alternative 3' splice site right on between the exon 2 and 3. So it may occur that they will not use this site. Instead of that, they use this 3' splice site for the splicing process, and due to this reason exon 1 directly binds with exon 3, whereas the intron and the exon number 2 may be spliced out. Okay, so this is the case that due to the presence of alternative 3' splice site as shown in this figure, the exon can bind with alternative way to the 5' splice site of exon number 1. So the exon number 1 of the 5' splice site, which is found on exon number 1, it can bind with the exon number 2 by the help of normal splicing process, or if they have a alternative 3' splice site, here it is showing right between the exon number 2 and 3, then exon number 1 can join with exon number 3, whereas the intron sequence between the exon number 1 and 2 as well as the exon number 2 can be spliced out during this process. So I hope you have understood. Again, I'm repeating, if you have not understood, this is the 5' splice site, this is the 3' splice site, this is an alternative 3' splice site. Now what can happen, exon number 1 can join with exon number 2 and this intron can be removed out by the process of splicing, because they have this 5' splice site, 3' splice site. So the spliceosome complex will interact with this site and will join the exons as just we know 1, 2 and 3. Or another thing can occur. The another thing is that an alternative 3' splice site is located right here and the 5' splice site is also located here. So exon number 1, so the spliceosome complex will can utilize this 3' splice site and 5' splice site, and this whole intron and exon number 2 can be spliced out. I hope now you have understood. Practically I have said, just in short words. Okay, example is also obviously it is given. We will go for the example. So an alternative 3' splice site junction called as acceptor site is used changing the 5 prime boundary of the downstream exon. So we are changing the 5 prime boundary, we are changing the boundary, and we are alternately we are trying to join with the help of that alternative 3 prime splice site. Best example of this type of alternative 3 prime splice site is the transformer activating protein, which is also called in short as TRA. This transformer activating protein present in the female of Drosophila melanogaster. Well, most of the alternative splicing um, experiments observe on this model organism of Drosophila. Okay, well, let us see how 3 prime splice alternative 3 prime splice site is being studied in Drosophila transformer activating protein. The original gene for the transformer activating protein contain two acceptor sites. That means, again I repeat, the gene responsible for the transformer activating protein, they have two 3 prime splice sites. So they have two 3 prime splice sites where the gene can split during transcription. So transformer activating protein is encoded by certain genes, and that gene have many exons, introns between them, and it is saying that they have two different acceptor sites. They have two different 3 prime splice sites where the genes can split during transcription process. Generally, in case of females, okay, the transformer activating protein is found in the females. Normally, male Drosophila uses the upstream acceptor site which contain the early stop codon. So there are two acceptor sites. There are two different 3 prime splice site. In case of male do not code for this transformer activating protein. Why male do not code for this transformer activating protein? Because male uses the upstream 3 prime splice site which contain an early stop codon. So there is a premature stop codon which will not code for that whole functional transformer activating protein. So transformer activating protein is not found in males. The gene is present, but as during the splicing process the male uses the upstream, that means towards the 5 prime in, there is a 3 prime splice site found in the males of that tra gene, transformer activating protein gene. So there is an early stop codon in that splice site, in that upstream acceptor site. That's why males will not code for that protein, and it forms a truncated or non-functional protein. Whereas in case of female uses the downstream acceptor site. That means towards the 3 prime in next, it uses the next 3 prime splice site. It uses the next 3 prime acceptor site, or you can say 3 prime splice site, which causes the stop codon to be skipped out, to be excised out, removed out as a part of the intron, forming a functional transformer activating protein. So with the help of diagram, maybe I can able to explain to you. Okay, so here it is saying that there is a gene called protein called tra protein. Okay, tra protein is what? Transformer activating protein. Now this tra protein is found in females. Okay, it is not found in males. Males do not code for this protein, but the mRNA is present. The mRNA from where the tra protein we get, right? It is found in both males and females. But why male does not code for this tra protein? Even a mRNA of the tra protein is present. It's because the mRNA have alternative 3 prime splice site. Well, if I say in case of males, this is the 5 prime, um, and suppose these are the intron exons. Okay, now exon number 1, 2, 3, how many exons are present? That's not the question here. The main question is why males do not code for this TRA protein. Okay, now it is saying that within the males, that the mRNA code for the TRA protein in case of males, within the upstream, it is saying that within the upstream, that means towards the 5 prime end, there is a 3 prime splice site, that is an upstream acceptor site, okay, which contains an early stop codon. Now within this splice site there is a stop codon present. So as usually when splicing occur, it will join with exon number 1, 2. Okay, that's good. That's okay, very good. But when from that mRNA protein is to be made, due to the presence of stop codon prematurely here, so the translation will process will continue up to here and it will stop. That's why functional TRA protein will not be made. But what happens in case of females skip this portion. Actually the females skip that portion. They don't use this 3 prime splice site here. They use an alternative 3 prime splice site. They use an alternative 3 prime splice site located right here. So they have to skip the whole part here. So the premature stop codon is skipped out. So no stop codon will be present in between them now, and thus they continue the splicing process, and from that splicing product that we get a functional mRNA. Okay, so premature stop codon is not present actually. I hope you have understood what I have said, that males have that mRNA, female have also have that mRNA, but male will not code for this TRA protein because in case of males, because they have two 3 prime splice sites, many 3 prime splice site is there, but one of the 3 prime splice site which is present in the upstream, it has a stop codon, and if splicing occurs in that portion, then the stop codon will be retained within the mRNA in case of male, and thus as stop codon is present prematurely, the translation process will stop and functional TRA activating protein will not be forming. But in case of female, they don't use that upstream 3 prime splice site. They don't use that upstream 3 prime acceptor site. They use this a downstream acceptor site where which will not involve the stop codon. The stop codon has to be skipped out, and thus as a part of the intron it will be skipped out, and thus we make a functional TRA protein after splicing. Okay, next is your alternative 5 prime donor site, where the donor site is the alternative 5 prime splice site, just like 3 prime splice site. Here the exons have alternative 5 prime splice site, so that they can bind with alternative way to the 3 prime splice site of exon number 3 as shown here, thus changing the mRNA transcript. So again I repeat, try to explain. So it is here from this figure, if you see, I have not mentioned the direction of the mRNA. So in alternative 5 prime splice site or alternative 5 prime donor site, as exons have different 5 prime splice site, so they can bind with alternative way. Like here you can see this is the 5 prime splice site, and this is another 5 prime splice site. This is the common 3 prime splice site, as you can see. So here either here they can interact with the exon number 1, okay, or they can able to interact with the exon number 2 here with that 5 prime splice site to make a um, different types of mRNA transcript. Like here in this case, if you see, if I try to draw in a, okay, so this is the 5 prime splice site, this is another 5 prime splice site, and this is the 3 prime splice site. Okay, so first case, in the first case what happened, as if they use this 5 prime splice site and 3 prime splice site for the splicing process, then what happened, exon number 2 will be skipped out, even the introns will be skipped out, exon number 1 and exon number 3 will remain. Okay, but if they use the exon number 2 and exon number 3 only, okay, then what will happen, whole of these exons will be joined because only the intron is removing now. Okay, so this is another form of alternative splicing where you can see that here the exons having an alternative 5 prime splice site, also called as donor site, so that they can bind with alternative way to the 3 prime splice site of exon number 3 as shown here, thus changing the whole mRNA transcript. Okay, now normally if I see both in case of alternative acceptor sites or donor sites, both of the sites can change in the protein's open reading frame, okay, and cause frame shift in the open reading frame. The last one is your intron retention. Now, simple thing, if you know the term what is retention, go through the dictionary. Retention means to retain, to remain. Okay, so here just we know in simple concept, splicing means to remove out the introns and join the exons. But here, sometimes what happened due to the absence of the 3 prime splice site, um, intron can remain within the mRNA. Now, if introns exist, they are often encoded in the coding regions among the amino acids of close, okay, by exons. The stop codon or a shift in the open reading frame will occur, which causes the proteins to become non-functional. So due to intron retention, as I have said earlier, that introns are non-coding portions, they do not code for proteins. So if introns did exist, they are often encoded within the coding regions among the amino acids of close by exons, and it may code for stop codon also, which or a shift in the open reading frame, which cause the protein to become non-functional. And this is least common mechanism in the alternative splicing. Okay, it is very found rarely. Intron retention is found very in rare case in case of alternative splicing. So this was all about the alternative splicing, where through a revision I will try to go. First is exon skipping, where you can skip any of the exons between them. It can be spliced out. Okay. The best example is Drosophila DSX gene. Mutually exclusive exons, all the exons will not remain together but in pairs. Okay, this is another way. Then I have said alternative 3 prime acceptor site, where there are alternative 3 prime splice site, which leads to different combination of mRNA spliced products. Best example of alternative 3 prime acceptor site is your TRA or transformer activating protein, which is found in females but not in males due to the presence of an stop codon in the upstream 3 prime splice site. Then 5 prime donor site, where the exons will have alternative 5 prime exons where they combine with the 3 prime splice site to make a different combination of products. Then the last one is the intron retention, where intron can remain after splicing. The intron can be included within the transcript, okay, within the mRNA after splicing. And as introns are non-coding regions, so it may cause change in it, not it will cause a shift in the open reading frame, and it can, due to the shifting of the open reading frame, a premature stop codon can occur, which can lead to non-functional protein product. So I hope you have understood this whole part regarding the types of alternative splicing. Just go through these notes, okay, that I have said. A short notes I have tried to prepare. In the next class, I will try to discuss about RNA editing. Okay, I have to finish off the genetics chapter now. The genetics part, like the flowering genes. Okay, so just go through these notes and ask if you have any doubts regarding this part that I have mentioned here today.